Thanks, Stephanie. Thanks, Hirmas and Michelle for organizing. Thanks, Hirmas, for hosting. It's really a pleasure to be here and an honor. Yes, I gave this talk a bit of a provocative title, maybe intentionally, but it will be. You'll see that this will be a little bit about brain structure and function as well. I'd like to set a little bit the context of the kind of work that I do. I think that in many respects, we are undergoing a phase shift in terms of the kind of research that we do in neuroscience by stepping into what I call the era of brain observatories. So the analogy that I think about are astronomy observatories. Observational astronomy moved from being kind of a cottage industry when individual researchers would do their own observations to becoming a centralized operation many years ago. And I think neuroscience is moving into that era as well. And some of the examples of that are projects like the Human Connectome Project that has collected a large number of samples. Other studies are more focused on sort of individual questions, like questions about aging, and the CAMCAN study that's also collected a large sample. Other samples focused on pediatric neuroimaging, like the Child Mind Institute's Healthy Brain Network. We'll come back to this one a little bit later, or the ABCD data set that both are collecting in the thousands, children, adolescents, in the case of ABCD also longitudinally. There are these kind of unique data sets that probe things that would be very, very hard for individual labs to study. The Developing Human Connectome Project focuses on fetal imaging and imaging in infants, newborn infants. Really unique project and very interesting data where we can ask questions that are very, very hard for individual labs to ask. And then the 500-pound gorilla of these kinds of approaches is the UK Biobank that's collecting data from 500,000 individuals. 100,000 of those will be also imaged using magnetic resonance imaging. So all of these projects provide very large data, and what I'd like to talk today about is the work that we're doing to address the challenges and maybe capitalize on the opportunities of the large data sets. So some of the challenges are very quotidian in nature. Some of the research procedures that we use just break when we use these very large data sets. Procedures that are routine in small data sets become close to impossible in large data sets. For example, detailed quality control. How do you look through the data of 100,000 scans in the way that you would look through just a smaller sample? Compute-intensive pipelines become hard to scale. But there are also interesting opportunities, machine learning algorithms, sophisticated machine learning algorithms that can capture complex nonlinear patterns in high-dimensional data. Those algorithms can provide really interesting insight into this kind of data, but they are notoriously quite data hungry and potentially sensitive to confounding factors. And to add, they can be opaque, hard to interpret. So that's a challenge as well. Oh, here's a black box from IKEA. To emphasize that. And then finally, a challenge that we have as a field is the challenge of education and training. Data science methods are increasingly available as open source software, but they often require specialized knowledge. In order to use the methods that I'll talk about today, sometimes you need to know how to program, how to think about machine learning algorithms, and so on. And this knowledge is rarely part of the curriculum that's being taught to neuroscientists, psychologists, medical providers, the people who do neuroscience. And researchers are not really properly incentivized often to develop, disseminate, maintain research software. It becomes a challenge to be able to do the kind of work that we need to do in order to be able to do data science. So what should we do with big data? Jim Gray, who was a computer scientist, the database researcher, kind of launched the field of data science. And that happened a little while ago. He kind of walked over to his colleagues in astronomy, and he worked with astronomers, also oceanographers. And he thought of this idea of the fourth paradigm. So the typical scientific archetypes are empiricism, experimental work, theory, building of theory, mathematical theory. Think of like Maxwell's equations, and computation. Think about the kinds of simulations that you can do in computers. And he said, the fourth paradigm is an entirely new phase that involves data-intensive practices, termed e-science. And this fourth paradigm, is very important to understand, doesn't supersede these previous paradigms, but augments it with new practices, new kinds of things that we can do. Indeed, nowadays we think of e-science as data-intensive discovery, data science, and the institute that I work at, called the E-Science Institute for historical reasons, because it takes its inspiration from these ideas from Jim Gray. My work focuses on the white matter, so I take these ideas to the study of the white matter. The white matter, I won't say much about it to this audience because you all know that it's important for many reasons. I think part of what's interesting about the white matter is that it seems to have a fixed plan of development. We know that there are waves of myelination that happen throughout life. We know that there's sort of a senescence that happens later in aging, that where there's demyelination. But in addition to this kind of fixed program of development, there is also a response to the environment. We know that the white matter is plastic. For example, this study where they showed that learning how to juggle changes the tissue properties of white matter in the parietal cortex. So it's a really interesting substrate, both for sort of development, but also for learning. And we know that it plays an important role in neural computation. And the way that I study this is using diffusion MRI data. Diffusion MRI is sensitive to the diffusion of water inside of each voxel in a directionally specific manner. That's done by applying magnetic field gradients across the sample, such that the image looks different if the gradients are, in this case, right to left relative to anterior, posterior. You can kind of see the color here changes when you change the gradient, and changes in a systematic way. And that's because the locations that we're looking at contain many axons, and the axons are also organized into an ultrastructure of fascicles, and the fascicles and the myelin and all that impedes the free motion of water in such a way that, as a caricature, the water is more likely to diffuse along the length of these tubes than across their boundaries. And we can capture that using a variety of models. This is the diffusion tensor imaging model. And we can model that to derive meaningful physical characteristics of the tissue. For example, mean diffusivity characterizes just how much the water can diffuse in all directions. And you can see that in the ventricles, there's a lot of diffusion, but in the white matter, adjacent to diffusion is a lot less. Fractional anisotropy is a good way to capture how different is the diffusion in different directions. And then we could find in which direction is there most diffusion, the principal diffusion direction. And that serves as a cue for computational track tracing or tractography. Here schematically, we're focusing in on a little box here, and we're fitting the diffusion tensor imaging model in every one of the voxels. And then if we place a seed in some location, we can propagate that by following the principal diffusion direction along the path of what we call a streamline. So we're propagating a streamline, that gives us one streamline. And maybe if we put the seed in another location close by and propagate that, maybe the streamline will go somewhere else. And if we repeat this process many, many times, we might get an image just like this one that contains millions of streamlines. So we get streamlines all throughout the white matter. Now, computational tractography has a variety of pitfalls, so we need to sort of think about what we're doing with it. There are inherent ambiguities in the diffusion signal. For example, here's an example from this paper from Meyer-Hein et al. From 2014. Let's assume that there is a crossing of fibers within this voxel. And this is the signal that we get. We get a signal that's consistent with a crossing. However, we could also assume kind of a bending or maybe a kissing or a crossing. So all of these configurations are consistent with the signal that's measured. And that's an inherent ambiguity that the tractography algorithms can fall into. Another pitfall is that there are larger uncertainty and there are certain systematic biases even close to cortex. This is a paper from Derek Jones from 2002, kind of quantifying the uncertainty. The paper didn't necessarily focus on cortex, but we know that this happens more wherever we have partial volumes of gray matter and white matter close to each other. The certainty about the direction of the diffusion becomes, get dispersion of the effects of the fibers on the diffusion. And this is a paper from Kurt Schilling demonstrating something that's known for a while now, that diffusion-based tractography has gyral bias. It tends to go to the gyrus, the crowns of the gyrus instead of the walls of the gyrus. And they demonstrate this very nicely in this paper, that even in cases where we know that there should be streamlines going to the walls of the gyrus, you still get this gyral bias. And then another kind of pitfall is that streamline counts are biased by shape and configuration. Here's another study by Derek Jones just demonstrating simulation that even if you set up a certain kind of configuration where the streamline counts should be uniform all over, you still get, depending on the shape and the crossing and other kinds of confounds, you get biased streamline counts in different places. So, well, before I'll tell you what to do with this, well, there is also good news. And the good news is that sentence here, brain connections derived from diffusion MRI tractography could be highly anatomically accurate if we know where white matter pathways start, where they end, and where they do not go. And that's the full title of this great paper by Kurt Schilling and his colleagues, I believe, in Brain Structure and Function. And so we can find these known white matter pathways, well-validated pathways that we know exist in every healthy human brain, and we know this from a variety of sources, both from measurements that are done in humans in vivo, but also from postmortem measurements and from comparisons to other species and so on. So these are kind of, we know there are real objects in the world that we can find in every individual using diffusion-based tractography. There's more good news, and this one I added a little bit late. I just thought the diffusion tensor imaging model has had a kind of there have been many challenges to that model. And I just wanted to emphasize that model, even though it's kind of the original, the OG model of diffusion MRI, it really is a good model still, even though people have developed many other models. It fits the data really very well. And this work that I did when I was a postdoc. And what we demonstrated in this paper is that if you take data, you fit the model, diffusion tensor model, to the data, and you cross-validate it on another observation, it fits the data better than test-retest reliability. That is, you're better off fitting the diffusion tensor model to the data than making another measurement of that. And so the data, and what this histogram shows is below this kind of number one means that the model, above the model cross-validates worse than the data, and below 1, the model cross-validates better than the data, and an overwhelming number of voxels or proportion of voxels in the white matter are in that regime where the model cross-validates better than the data. So the models are really accurate. So what can we measure with diffusion MRI, given all that? Well, think about an analogy of some city that you're in, and you're trying to understand that city and what's going on in the city, and this is based on an analogy by Colby and colleagues that was published in 2012. You're trying to see what is the topography of the city, what's the traffic like. So let's assume that I'm in the lodge over here, and I'm trying to get to here, which is where the blue dot is right there. And I want to know what the situation is between here and there. So maybe that's not something that I can do with the methods that I have. Instead, maybe I need to zoom out maybe even more. So maybe what I can do is I can focus on the highways and information about these highways and maybe the topography in sort of a broad scale. And that's still useful information to know. So even if we can't know the connectivity from some point on the gray matter to some other point next door, we can still get a lot of information about things that are quite interesting to study. And so, in our approach, we capitalize on these advantages and try to avoid the pitfalls as much as we can. We've been working with Jason Yateman, who invented this method called automated fiber quantification. In this method, we find these well-known validated tracts, and we characterize their tissue properties along their lengths using models like diffusion tensor imaging and other related models. And so what we get out of that is we get, for every major tract, we get this tract profile. So this is the fractional anisotropy along bundle, in this case, the arcuate fasciculus, along its entire length. And you can see that it varies in a systematic way, and we can characterize this in every individual. And I should say, John Kruper, who's a graduate student in my group, has written software called PyAFQ that does this automatically with data sets. And we really put a lot of effort into open source software development so that others can use these methods as well. This method is robust to methodological choices. It's also got high test-retest reliability, as John demonstrated in the paper that we wrote in 2021. So this is a pretty good approach to sort of take into these large data sets. So here's an example of a few of these white matter tracts found in an individual from the Human Connectome Project. And this is work that Mackenzie Hagan, John, and Asa Gilmer, students in my group, have done on this data set. Here are the track profiles, the average track profiles from all of the Human Connectome Project, in this case for FA, and here for mean diffusivity. And we've even created a little viewer that you can go and look at all of the data from the Human Connectome Project. This is work that Asa did. We call it Tractoscope, which thought was a really clever name, and you can find it at that website. We also released all of this data on Amazon Web Services. So all of the tractograms, all of the bundles, all of the track profiles are available through this, the open data program from Amazon Web Services. And I also point out Azure there because computing this large scale data sets, it takes a lot of compute, and we had to use these cloud computing resources in order to do that, and we leaned on Azure and Amazon Web Services in order to do that. And we developed tools in order to be able to do that more rapidly and in a more reproducible manner as well. Okay, so let's talk about research procedures that become challenging with large data sets. So pretty early on when we started thinking about what we want to do with these tools is we wanted to study, because Jason is very interested in reading, wanted to study these pediatric data sets and understand reading ability and how it manifests in these data sets. And one of the problems with these data sets is data quality. Children tend to move in the scanner. And so Anisha Keshavan, who was a postdoc with me and with Jason at the University of Washington, tried to solve this. This is not diffusion MRI, this is T1 weighted imaging, and she focused first on T1 weighted MRIs. Anisha, in her PhD, she had developed tools for quality control by experts. So we took some of the data from the Healthy Brain Network, of the T1 weighted, and we had a team of experts look at it meticulously and do the kind of standard quality control that we usually do. But the problem with that is it doesn't scale to these very large data sets. We can't do that for 2,000 brains. We could do it for 200 brains, but it's very hard to do for 2,000. So what she did is she decided to engage the community and built a community science app. This is an app that lives on a website, brainder.us. And it is, you see a slice of a brain and you decide, swipe to left if it doesn't look good, swipe to the right if it does look good. Inspired, of course, by Tinder, the dating app. And we actually got some traction, and she tweeted it out. People got interested. It was kind of fun to do because it's kind of like this kind of task that you can do on the bus or while you're waiting for the bus. But the problem is we got pretty inconsistent ratings from different individuals. So, some people, they look at this and they know what they're doing, and other people look at this and they don't know what they're doing. So we get lots of data with a variety of qualities. And so what Anisha did is she took the gold standard data set that we had from the experts, and she designed a model that would differentiate people who are more like the experts from people who are less like the experts. And so now we could weight their contributions proportionally. So some people were really good and they got weighted very high. Other people were not as good at this or not as consistent with the experts, and we down-weighted their contributions, and we got much more reliable and consistent ratings doing that. In fact, we also got a better match to the experts. So these are ROC curves that show that if we just take the average that we got from the community scientists without considering their match to the experts, we get an ROC curve of 0.95. That's already pretty good, actually. But if we do train one of these algorithms, we can go a lot higher with that. Now, one of the aspects of this work that then are I think, interesting and useful is that we can take the data that we've now harvested from the community, and we can train a new algorithm that we can then apply to subsequent data as well. So this is the full game here. We had the experts rate a gold standard set. We had community scientists rate using the app, and then we trained a neural network using the data that we got from the community scientists. And that's because these algorithms require lots of data. And then we got an algorithm that matches experts really, really well. The area under the curve is about 0.99 for this neural network. And this neural network never gets tired. This neural network can be used on subsequent data collection because data collection in the Healthy Brain Network is still ongoing. So we can apply it forward to future data collection as well. And then Anisha did one more move, which is to take this idea and generalize it into an open source software project called Swipes for Science, where people can build these kinds of apps for a variety of different kinds of data. In fact, others have taken it, Damien Fair and his group have taken it now and used that Swipes for Science idea in some of the work that they're doing with ABCD for quality control, and also they're applying it to the new Healthy Brain and Child Development study, which will go to even younger ages. A really interesting study. I took a shot of Damien's talk at OHBM showing Swipes for Science being used in HBCD. So we're really excited that this idea, because we developed the software and launched down to the world, it's easy for others to take this idea and pick it up and run with it. Okay, so then once we had this idea in place, we thought, well, now we can move on to the diffusion MRI, and that was work that Adam Ritchie-Halford, who was a postdoc with me, did, and we collaborated with Matt Cieslak, and he's in Ted Satterwhite's group at UPenn. And we developed fiber.dev, very similar idea, swiping app, this time for quality control on diffusion MRI data, and that's a representation of diffusion MRI data that we used. We used kind of like the non-diffusion data as the background, and then these colored FA maps as the foreground. And what we had is kind of, you can see this kind of data pipeline here. We started with raw data from about, well, 2,500 subjects. We did some standardization, pre-processing. We did expert QC on 200 subjects. And then community scientists rated a large number in addition, about 1,500 or so. And then we had the neural network apply to that as well. And we got accuracy. It wasn't quite as high as the T1 weighted, about 0.95, but still quite useful. We had to develop along the way all kinds of tools for computing on this very large data set. We have CloudNod is a software that automates deployment of these kind of pipelines to Amazon's web services. And we ran all this through Amazon's web service. Okay, so here's the track profiles then from the healthy brain network data set. One thing you can notice, so these are colored, and what we did is here we divided the sample up into bins of 3 years in age. I believe the youngest child here is 5 years old, and the oldest is 21 years old. And what you can see is a very consistent increase in FA that is very broadly in the entire brain. The brain really changes throughout childhood and adolescence. No surprise there. In mean diffusivity, we see kind of the opposite pattern. Mean diffusivity decreases, and that's also, it's a very global pattern. You see it everywhere. And what we wanted to do is we wanted to build models that can kind of assess brain age. But once we have this kind of pattern, we think, well, maybe we can build an algorithm that predicts the age of an individual based on their diffusion data. It's a common benchmark in human neuroimaging. And these models are also useful because if somebody deviates from the prediction, for example, you have a child and their predicted age is younger than their actual chronological age, you can look and see whether that indicates something about that person. You can also say, well, maybe this person's brain age looks higher than their actual age. That's also informative. And what we realized is that tractometry is a form of feature engineering. In machine learning, people talk about feature engineering as the process of going from some kind of raw data to data that we can do machine learning with. So it's, in this case, it's tabular data. Each column here is a particular point along the length of a particular tract. Schematically, for example, all the corticospinal tract is here, and all the cingulum bundle is here, and all the arcuate is here. And we can set this up as a big linear model, a multilinear model, and we can do statistics on that. We were helped by Noah Simon, who was my colleague in biostatistics, and developed this toolbox, AFQ Insight, that does machine learning on track profiles. So for example, you can predict the age of an individual based on solving this kind of big set of equations for beta here. And we used an algorithm that Noah developed as a statistical approach Noah developed called sparse group lasso, and that works really well in the healthy brain network in this paper that Adam published. But the question is, can we do better if we use a nonlinear approach? And we wanted to use new deep learning algorithms. So this is an experiment, a computational experiment that we did where we wanted to see how data hungry are these algorithms. On the x-axis here is the number of training samples. On the y-axis is the accuracy, the R squared, or variance explained by this kind of brain prediction algorithm in cross-validation. And what we can see, this PCR lasso is an approach very similar to the one that I just showed you. It's a kind of linear model. But then we tried several different kinds of nonlinear deep learning algorithms, and we can see that there is actually a benefit. It's not very big because this linear model is already very, very accurate, but there is a benefit to using patterns that are not apparent when you use linear models. So that's pretty good news. Wanted to try to apply that idea to some interesting questions beyond development. So here we're looking actually at aging. In this case, we're looking at retinal disease. We're looking at glaucoma. Glaucoma is the most common cause of blindness. And in the UK Biobank, we can ask a set of interesting questions about retinal disease and its relationship to the white matter. The kind of question we're interested in is, does change to the sensory periphery, glaucoma in the retina, correspond to change in the white matter? And here's the kind of sample we're working with here. So these are, I believe, yeah, something like 7,000 or 6,000 in some subjects from UK Biobank. And in green, we've colored the individuals who have glaucoma. In red are healthy controls. One of the problems that we immediately see here is that the glaucoma tends to happen in older ages. And so what we did when we have this kind of large sample, one thing that we can do that we think is a neat trick is we can match the group of interest, in this case people who have glaucoma, with a set of very well-matched controls. So in this case, we matched on age, sex, and socioeconomic status. So each individual in the glaucoma group had, from this group of more than 5,000 individuals, had selected the best match on age, sex, and socioeconomic status so that we could get rid of confounds. Part of the problem with these deep learning algorithms, they'll pick up information from wherever they can. So if we had given them this data, they would have immediately picked up the age information, which is very apparent in this data, and just run with it. By doing this, we neutralize the age information because just knowing the age of the individual doesn't tell you whether they have glaucoma. Instead, you have to focus on the information from the track profile. And we focused on, in this case, we focused on the optic radiation. So this image from a review article that Hiramasa and I recently wrote, was recently published actually, shows kind of the process of getting the track profile, in this case, from the optic radiations. This is a young child from the Healthy Brain Network, but the same idea works also in the UK Biobank. So what John did with this data is he kind of plotted the track profiles. And what you'll see is that there is a difference between people with the controls and the glaucoma, people with glaucoma in the sample. I should say the solid line is the average, and then these thin lines hugging the solid line, those are the 95% confidence intervals. These very thin lines on the edges are the interquartile ranges. So on the one hand, the confidence interval is very, very small, and there's clearly a significant difference. On the other hand, the interquartile ranges are very large. We don't see that in control bundles, in this case in the corticospinal tract. So in theory, this is really, really statistically significant. But as my colleague Daniela Witten, who is, I think, one of the leading statisticians of her generation, said in this very cogent Twitter rant, a p-value is just a test of sample size. Given a certain effect size, if you just increase your sample size very, very large, you'll get a statistically significant result. In this case, the sample is very large, and so maybe the effect is significant, but does it have any practical significance? And in order to test that, again, we go to machine learning, and we try to fit these kind of models that I showed you before for predicting age, but to the question of classifying individuals with glaucoma. And sort of the question is, by looking at their white matter in the optic radiations, far away from the eyes, but not that far, can we tell who has glaucoma and who does not have glaucoma? The answer is that using a linear model, we can't do that very well. And the reason for that is because of this overlap that I showed you over here. There's just a large overlap between these populations. And we definitely can't do it using these control bundles. The question is, what about a deep learning approach? And with a deep learning approach, we can do a little better, let's say. So, an area under the curve of 0.69 is not spectacular. This is not a diagnostic tool, but it does indicate that there is some information in the track profiles that differentiates individuals with glaucoma from individuals who do not have glaucoma. And this comparison tells us that that information is not apparent immediately by looking just at a linear comparison. You have to do a little bit more to pull that information out. Moreover, the model does not generalize the discrimination between younger and older subjects. So we don't think of this as an indication of some signal that is more rapid aging, maybe, of that part of the brain. It also does not generalize the classification of age-related macular degeneration. So we don't think of this model as a model of just the visual input has changed. It's something that has to do with glaucoma. Well, we only tried age-related macular degeneration. So that's all I can say. It does not generalize to that. But we still have a black box in our hands, which is a problem. And so in order to kind of try to tackle that problem, we tried to reach out to methods from the machine learning community for opening up the black box. So Scott Lundberg, Suin Lee is in the computer science department at the University of Washington, and they had developed a set. Suin is an expert on what's called explainable AI. These are methods to take these black box models and open them up in order to look at what they're doing and how they're doing what they're doing. Scott, in particular, when he was a PhD student in her group, developed a set of methods based on game theoretical math. He called it SHAP because Shapley additive explanations is kind of the term from the math and the game theory. So the software is called SHAP and the method is called SHAP. An intuition about this kind of method is that it takes the predictions from the deep learning network, and for every subject, it fits a locally linear model. So you take the subject's data and you say, what caused you to say linear deep learning network? What made you say that this person has glaucoma? And you can do that by fitting a locally linear model on all the features and see which of the features really drives the prediction on kind of a locally linear subset of the data. This is I should say, just to be fair, that this is a simplification of the method, but it's enough in order to understand what it does. And so for each subject, this method identifies the data features that most contribute to their classification. Let me give you an example. This is actually work that we published a few years ago, looking at the retinal imaging from the UK Biobank and some of the kind of diagnostic or phenotypical data that exists with the UK Biobank. This is work that Permita Mehta, who's a computer science student at the University of Washington, did. We collaborated with Suin and with others there in order to do this. And what you can see is, so here are three different kind of features. The orange dots here are individuals with glaucoma. Green are individuals who do not have glaucoma. And the way to read this is on the x-axis is one of these features, for example, intraocular pressure. And if the y-axis, the value on the y-axis, the SHAP value is high, then that means that for that individual, the intraocular pressure was a strongly indicative feature for that person having glaucoma. So for example, there are some individuals out here that are healthy, but their intraocular pressure is very high, and the algorithm really thinks that they should have glaucoma based on that. Would have decided if it only were deciding based on this that they do have glaucoma. Age has sort of an interesting structure. Young people, negative values. So negative values in this case is a strong indication that their age indicates that they do not have glaucoma. But as they age, there's these interesting step functions here at 55 and 65 years of age. We can speculate why that is. And then an interesting, so these are features that were known, but forced vital capacity, which is a lung function feature, that was not something that was necessarily known to be indicative of glaucoma, and it is strongly indicative of glaucoma, especially when people have very low forced vital capacity. That's sort of something that we discovered using this method. Okay, and then we took this method and John applied it to the track profiles. Here, the way to read this is on the x-axis is the value of, for example, in this panel, the fractional anisotropy, and it's normalized such that high values here are sort of relatively high values of FA, and low or negative values here are relatively low values of FA. You could think of it as a z-score relative to the distribution. And then on the y-axis is the value of the SHAP kind of averaged across all the individuals that are sort of in, or all the values actually across the entire data set that are in that bin. So one thing to see is like very low values of FA are in the optic gradation are very indicative of glaucoma because this is positive up. And high values are weakly indicative of no glaucoma. Interestingly, mean diffusivity has a very, very similar pattern, although we might have expected that if this was going along with aging, that this would be the inverse, actually. But it's not. Mean kurtosis also has, the mean kurtosis of the tensor also has an interesting kind of nonlinear pattern here, where extreme values are indicative of glaucoma. So this gives us a little bit more of a handle, and these, I should say, are sort of preliminary results using this approach, a little bit more of a handle on what these deep learning algorithms do, and I think quite promising in terms of how we might use these algorithms in future use cases. Okay, let me finish with just one thing. I mentioned in the beginning of the talk that, well, there are all these methods and we develop them, and it takes a lot of knowledge to be able to use these tools, and this is not knowledge that we naturally I think, give to neuroscience students in their training, or we're just struggling to give, I should say. These data science methods require specialized knowledge, like software engineering, machine learning, high-performance computing, and many other things. The question is, how can we teach all this? So I'd like to offer two solutions that I've worked on. The first is this thing called Neurocademy. Neurocademy is a summer school in neuroimaging and data science. We teach all of those things that I mentioned, and then we do that in the first week. And then in the second week, the participants work on a variety of hack projects. It looks like this. They get frustrated. They get really happy. And Tal Yarkoni is up there because he started that with me. I'll finish in just a minute. We started this. We're doing it for the 9th time, just in a couple of weeks, in a few weeks, end of this month. And then Tal retired from science and is doing other things now. And Noah Benson, my colleague, has taken over that. And he's my collaborator in this. And it's been really fun to work with him. And if you're a student sitting in the audience thinking, oh, well, I can't do this year because it's just in a few weeks, rest assured that we are funded to do this through 2027 at least. So you'll have several more opportunities to do this. One more last thing here is Tal and I wrote also a book called Data Science for Neuroimaging that came out late last year and also talks about all these methods, try to teach people how to do all these methods. The content of the book is also available on that website, so you don't necessarily have to buy it if you're just interested in the content, but please do buy it. No, just kidding, you can buy it if you want. And with that, I'll finish. I just need to give thanks to all my funding agencies and then, of course, to members of my lab here on the right. And just one more plug is that if you're interested in trichtography, there was recently the Maxime de Couteau and others have recently launched an international society for trichtography. And this is aimed at all different levels of trichtography. So if you're working on measurements in microscopy, of histology, and brain connections at that level, or if you're working at the level like I am of computational trichtography and neuroimaging, this society is aimed to kind of bridge these different scales and foster interdisciplinary conversations, and you can now become a member and follow that society. So I thought I'd give it a plug as well here so you know about it. And with that, I'll stop and take any questions.